In January 2015, experts in cancer research from around the world gathered at the RIKEN Yokohama campus in Japan. They were there to take part in a special meeting organised by the EC-funded MODHEP project, probably one of the most comprehensive research efforts ever undertaken to describe the mechanisms behind liver cancer, particularly hepatocellular carcinoma, or HCC, one of the most common malignancies worldwide, and in essence, still an incurable disease. RIKEN plays a pivotal role in the project, bringing to the table its expertise in the computational analysis, known as cage technology, needed to understand the complex datasets being produced during experimentation by the labs of its MODHEP partners based throughout Europe. The 5-day meeting in Japan had 3 main objectives: to offer special training to up-and-coming young scientists involved in cancer research of the cage technology devised at RIKEN, to provide a platform for discussing our understanding of liver cancer development at every level at a global symposium, and to host the 4th annual MODHEP project meeting, during which partners were able to discuss the progress being made, the implications of their work in relation to the overall aims, and to plan for the next stages for the remaining year and a half.
Is an in-depth, informal scientific discussion with critical insight of where we are and where we go from today till the end of the project.
MODHEP is an EU project funded through FP7. It is made up of 17 partner organisations from 8 different countries, 5 EU member states, Israel, Australia and Japan. The €12 million project started in 2011 and will last 5.5 years. The MODHEP consortium brings together world-class scientists and researchers with wide-ranging expertise, ensuring it has the skills required to achieve its ambitious aims and objectives. These are to provide genome-wide understanding of alterations occurring during initiation and progression of liver cancer, analysis of the genetic, epigenetic, higher-order chromatin and nuclear architecture, and to integrate all of these features into a systems-based model of the disease. It was this work that was under scrutiny during the Global Symposium and the MODHEP annual meeting in Japan, and as project coordinator, Bruno Amati explains, the objective was to gain a real understanding of the progress being made in all the different MODHEP labs involved in the project, and how this can be brought together to paint a holistic scientific picture.
We all know that cancer is a genetic disease in the sense that it's driven by mutations in the genome that eventually lead to changes in gene expression that characterize and drive tumor cells. Right? There's a number of levels of organization of the genome which go wrong. And MODHEP has come together to try to study how these different levels of organization of the genome and the regulation of the genome go wrong. We have specialists in various different levels of genome organization and gene regulation. All of these specialists work on the same material, on the same tumor models from the mouse and also on clinical samples. And they apply their level, their know-how and their specific level of analysis to the same samples. This is being integrated into the global picture, which actually would show how the system is organized and which are the weak points on which intervention would be possible.
A combined understanding of the different levels of genome organization and gene regulation is one of the key strengths of MODHEP. Wouter de Laat of the Hubrecht Institute has been looking at the 3-dimensional organization of the genome within the nucleus.
In principle, we have 2 meters of DNA if you completely unfold it in every tiny cell nucleus, and the folding of which is very intricate because it needs to be packed in this tiny cell nucleus, and in the folding is a lot of function of the genome hidden. So in the end, it matters which pieces of DNA from elsewhere on the chromosome actually physically are together in the nuclear space. And this 3C type of technology is a biochemical method by which you can capture the 3D organization of our DNA, of our genome inside the cell nucleus. So that these technologies enable us to understand if a gene starts expressing wrongly, so you start to make too much product of this gene, it can be the consequence of an important regulatory piece of DNA elsewhere on the chromosome physically being in touch with this gene and causing it to be transcribed much more efficiently than that it would normally do. So if we want to understand cancer and how genes are misregulated, it's important to also implicate the three-dimensional organization of the genome and to try to understand why certain genes go up or down in their expression. So that is, I guess, one of the novel angles of MODHEP.
Meanwhile, Yu Wei of the Institut Pasteur in Paris has been working on the functional validation of candidate genes.
So after the high-throughput screening of tumor samples, we can identify genes up or down-regulated in tumor samples compared to non-tumor samples. So these genes can be either driver genes in the carcinogenesis or just passenger genes, which means there's no really significant roles in carcinogenesis. So it's important to validate which gene is a driver gene in carcinogenesis and which gene is not.
Combining the data from all the MODHEP work packages into an integrated model is an essential step in creating a global picture of liver cancer.
And only when you start integrating these data, so that is actually one of the major efforts from the consortium. Everybody is collecting these huge data sets, but they only start to make sense if we start integrating them, and then we can get the full picture epigenetically of how cancer proceeds, and perhaps find ways to where we can try to interfere with that process.
Valerio Bianchi of the Italian Institute of Technology is involved in the computational side of MODHEP that is so important for analyzing the large data sets produced by the other work packages in the project.
Right now we need big data because there was a revolution in the last 10 years in which we passed from studying the single gene and how these affect, for example, the progression in cancer cell development. But then we discovered that usually a cancer is a very complex system in which you don't have only a single gene that is affecting the system, but you have a bunch of genes, maybe many genes. In this case, the role of bioinformatics is to identify correlation between data and extract a new high level of information to prepare a new set of experiments and then permit to the wet biologists to go further with their experiment. Bioinformatics, in general computational biology, can create predictors that, starting from the data that you obtain with next generation sequencing technologies, you can start to create predictors that try to identify which are the main characteristics of tumors. Can we predict if we're using, for example, a drug, if we are able to replicate the tumor, how the tumor change the state of the genes, and if we are able to do that, can we ask, so do we have a drug that can repress these genes in order to revert the tumor progression?
Computational analysis has in recent years become an indispensable skill for all scientists. The MODHEP consortium has responded to this and organized a cage technology training day during the meeting in Japan.
So this training course was designed for experimentalists, because now experimentalists also need to analyze a lot of data. So we try to provide a basic idea what is the cage technology, what is the phantom project, and also how to use basic tools to analyze sequence data or large-scale data.
We are here in Japan, hosted by one of our partners, Piero Carninci, who has great expertise in the analysis of gene expression with very specific techniques that have been developed here. Very advanced techniques that allow to map genome-wide the beginning of all the genes that are active in the genome.
I like to develop technologies, in particular develop technologies to analyze the transcriptome, which is the part of the genome that is expressed in RNAs. Yes, we have been specializing in over the years to develop a technology to capture the RNA from their cap, so that is a part of the molecule that can easily be used to select the part of RNA that is very very close to the initiation site. So with this technology, which is called CAGE, we can map in a high throughput fashion all the promoters that are active in a given instance in all the cells that have been analyzed, and the amount of RNA that is produced at each promoter. This analysis is very helpful to understand gene regulation, gene networks. Very important also, we have been developing computational tools to analyze those data, so we can we do not only produce sequences, but we also have tools to analyze.
These experiments and analyses have been brought together through a combination of innovative systems biology-based approaches that will provide a model of the development of liver cancer. Giuseppe Testa of the European Institute of Oncology and member of the MODHEP Ethics and Scientific Advisory Board explains the significance of this approach for our understanding of all cancers.
We are, opening up at a level of resolution and so of precision that was really never achieved before, the steps that lead a normal liver cell to become a tumor liver cell. And of course, this is fundamental for any cancer. So here, liver is just one, but very important example. And so I think that really MODHEP will pave the way to, to a cutting-edge approach to understand the history of cancer. I mean, cancer, like many of the diseases that are still very hard to cure, are very hard to cure, also because we arrive, so to say, when the crime has already been committed. We arrive late. And we arrive late because, for obvious reasons, we really almost never had the option of studying how the disease unfolds, how it develops. MODHEP uses some very interesting models which basically enable precisely that. So to follow the various steps that it takes to go from a normal cell to a cancer cell, in this case in the liver, but the results could be interestingly translated hopefully also to other tumors, and thereby set a paradigm for what kind of knowledge you need to acquire and importantly integrate in order to actually navigate these transitions. Of course, with a view to then eventually use this data to define key molecules that one day will be targets of new drugs or existing drugs to actually remedy this disease.
Of course, we are in a very specific, we're dealing with specific aspects of one very specific type of cancer. But this is how research is organized, and this is how we add, bit by bit, the knowledge that will allow eventually to develop cures against all these different diseases.